Good morning, everyone. We welcome you once again to our CONALab Talks. And today, of course, is about the water testing series and on which we will discuss drinking water specifically. So my name is Aletia Cruz, and I am a microbiology product manager here at CONALab. So let's have fun and let's have a good time and let's begin. So if this is your first time using this platform, so just to comment quickly, we have a box. So wait a second. Now I'm sharing my screen, of course. So if this is your first time using this platform, you have this box through which you can send us your questions or doubts that arise throughout the presentation, and at the end we will take a couple of minutes to answer them. Today's presentation is mainly divided into 5 main parts. So a bit of context on drinking water, a review on the membrane filtration method, followed by the ISO methods required to perform in this type of water. Of course, alternative methods for these analysis, and finally, just a quick review of the Condachrome solutions and the available products. So let's get to it. Let's start with the importance of drinking water quality. So water is, of course, essential to maintaining healthy and prosperous communities. However, a large part of the population takes this access for granted. It has been estimated that 71% of the population has access to a safe drinking water service, according to the WHO definition, the World Health Organization, of course. This definition is one where water is available when required, that is, of course, at home, and is free from contamination. However, this means that almost a third of the world's population does not have a safe access to drinking water. And this has, of course, a direct negative effect on health because it may contain microbiological, chemical, and even radioactive contaminants. When there is contamination with human and animal feces, water contributes to waterborne diseases that are infections that can lead to most commonly gastrointestinal problems or even systemic diseases, also diseases caused by water. And these ones are related to hygiene due to a poor access to drinking water. Aquatic diseases caused by worms that live in the aquatic fauna, and also water-related diseases. These are caused by insects that reproduce in water, and we can find them in, for example, malaria and dengue. So in this webinar, we will focus on the former, since 1.5 million people die of waterborne diseases annually. The vast majority, unfortunately, are children who die of dehydration caused by diarrhea. So it is not surprising that water is one of the main vectors of disease. Since the 19th century, the transmission of pathogens through water was established, followed by a cholera epidemic that resulted in 500 deaths. And then an English doctor and epidemiologist, of course, noted a relationship between the cholera epidemic and the deaths that occurred. He isolated the responsible bacteria and established the first link between the consumption of contaminated water and the outbreak of the disease. Then, in the 20th century, cholera and typhoid were the main diseases transmitted by drinking water. And since then, others have followed, such, of course, as Campylobacter, Salmonella, Listeria, Vibrio. Specifically, in the European Union in 2019, there were waterborne outbreaks related to STEC, Campylobacter, Giardia, and some other viruses, such as norovirus. The contaminated source was tap and well water. So the most common route of infection is through the fecal-oral route, meaning that the infection occurs as a result of exposure to water or food treated with sewage or directly contaminated water with human or animal feces. These microorganisms are known as the enteric pathogens that have historically been the focus of diseases transmitted through water, mainly because, as we have mentioned before, they were the first to be detected, and also because partly they are the main responsible for large epidemics such as cholera, typhoid, and dysentery. Other microorganisms, of course, can cause diseases through water of non-fecal origin. And these are derived from the consumption or use of drinking water, since they are autochthonous microorganisms from the water source or even the environment. For example, Legionella, which is transmitted through aerosols, is a frequent cause of diseases associated with drinking water. And that is why the surveillance and monitoring of this bacterium began. The main categories of waterborne pathogens are bacteria, usually enteric like Salmonella, Shigella, Vibrio, E. coli, and others like Legionella, Mycobacterium that are aquatic bacteria. They are relatively easy to remove through the established wastewater processes and are more or less a hazard depending on the country. Viruses, these are released in the aquatic environment and are present in fecal matter only if the individual or animal is infected. And while enteric viruses do exist, they differ quite a bit from the enteric bacteria in that they cannot multiply outside of their host cell and can only specifically infect certain species. However, they can persist in the environment for longer periods than would be like bacteria. Parasites are a diverse group of organisms with the common characteristic that they live in or on a host organism and obtain their food from or at the expense of it. They are classified into two groups, protozoa and helminths. The first are released in aquatic environments as cysts and are quite resistant to environmental conditions. And the latter include, for example, intestinal worms. Their danger lies in the duration of the diseases they can cause, as well as their high resistance to disinfectant substances. And since there are a wide variety of organisms that can contaminate drinking water, testing for all these types of pathogens is neither feasible nor realistic. Therefore, as a consequence, the monitoring has been carried out using certain microbial indicators to ensure that the quality of these waters is safe. The rationale is very simple. If it's found in water, it must contain fecal matter and therefore also contain pathogenic microorganisms. Common indicators or group of indicators are, of course, the known famous total coliform bacteria and can also be others such as thermotolerant coliform bacteria. This is why the European Union has Directive 98/83 on the quality of water intended for human consumption. At a national level, this has been reflected in Royal Decree 140/2003, in which these microbiological criteria are also established. This legislation was enacted several years ago, so obviously there have been modifications. However, given research and technologies advanced at such a degree that now it has been necessary to substantially modify this directive. And this has resulted as a new directive that was published at the end of 2020. This is Directive 2020-2184, and it repeals the previous one, and it must be adopted before January 13th, 2023, so we have a little bit over a year to do so. In this slide, we can see the microbiological parameters that are indicated in each directive and that are mostly maintained, such as E. coli, coliforms, of course, enteric bacteria, enterococci, Clostridium. In circumstances where it is recommended by the risk assessment, Pseudomonas aeruginosa is eliminated and, if you will, replaced with this new addition in the microbial control. There are the somatic colifages that also, as Clostridium, will be measured if the risk assessment indicates it appropriate. So coliphages have proven to be very interesting, and it is that because viruses have been shown to be the main culprits, even if we wouldn't think so, of gastrointestinal diseases. The problem is that the common disinfection treatments are not always as effective at removing them. And again, we focus on enteric viruses, which are those that we can find in contamination from human and animal sources, specifically a subgroup of these that selectively infect E. coli cells known as coliphages. These have been proposed as a more reliable indicator than, for example, enteric bacteria. For these reasons, their persistence is greater in the environment. They reproduce in a controlled way in the presence, of course, of E. coli host cells, and their resistance to physical and chemical treatments currently used. So those are the three main points, because that are the reason these have been deemed as a very reliable indicator. So now, in the second part of the webinar, we will quickly review one of the most used techniques in water analysis, which is, of course, the membrane filtration method. For this, we have ISO 8199, where the techniques for the detection and enumeration of bacteria, molds, and yeasts are indicated to evaluate the quality of the water. In counting methods, there are three techniques: the method of membrane filtration, pour plate technique, and spread plate technique. So in general, for any of these methods, the sample is inoculated into a solid or a molten culture medium so that the microorganisms of interest form colonies on or in the medium. The result is, of course, expressed as colony-forming units per sample volume. So now about the membrane filtration technique. You begin by positioning the sterile funnel, which would be this one, over the flask and holding it securely. Then with sterile flat-tipped forceps, the filter, which would be this one, is placed on the base, which means the porous plate of the filtration apparatus, with the grid facing up. And then the funnel, of course, is placed again. The sample is aseptically transferred into the funnel, and the stopcock of the is opened to apply the vacuum and thus filter the sample. This is when the sample will be passing from the funnel to the flask. And once it has been filtered, we close again the stopcock to seize the vacuum. So according to the ISO, it is very advisable to rinse the funnel with diluent to eliminate any microorganisms that may have added to the funnel, to the surface of the funnel, of course. Once the filtration is finished, we go on to lift the funnel with care, and then with the help of the edge of the base of the filtration apparatus, the filter is removed with the forceps and the membrane is carefully transferred onto a Petri dish. It can be usually placed on the surface of an agar plate with the grid facing up, and extreme care must be taken to place the membrane flat on the surface of the agar, so we avoid any forming air bubbles between the agar and the membrane. So molten agar can also be used to cover the filter with the medium, and well, the inoculated plates are then incubated. You can use an incubator or a water bath, and of course, in the plate in an airtight container in the water bath. And the incubation conditions will depend on the microorganisms of interest. So a single filtration apparatus or a manifold system can also be used for simultaneous filtration of multiple samples, which would prove very useful when the volume of analysis is important. So let's quickly review all the analysis that must be carried out for drinking water. Going back again to the current directive, 9883, we have that the parameters are E. coli, enterococci, Pseudomonas aeruginosa, colony count at 22 and 37 degrees Celsius, and Clostridium perfringens, depending on the situation, of course. So let's get right into it. So we have ISO 9308-1 for the enumeration of E. coli and coliforms by membrane filtration technique. So it is very simple, this method. A portion of the sample is filtered using the, again, membrane filtration method, and the filter is placed on a CCA plate. That is a chromogenic medium that will allow simple differentiation through the different pigmentation that the cells will present. It is very important to mention that coliform bacteria, being members of Enterobacteriaceae, express the enzyme beta-D galactosidase, while E. coli, also a member of this family, expresses beta-D galactosidase, but also the enzyme beta-D glucuronidase, which is not present in other coliforms. Then this beta-D galactosidase enzyme, by degrading the chromogenic substrate of the medium, generates colonies of a pink color. And E. coli have it not only galactosidase, but also glucuronidase. The colonies will grow purple or dark blue due to the joint action of these two enzymes with the chromogenic substances. This can be clearly seen in this image we have here on the right. On the left, we have growth of E. coli, and on the right side would be colonies of coliforms. So the filter is incubated and then examined. Pink-red colonies are listed as suspected coliforms, and blue-violet colonies as suspected E. coli. The total count would be all colonies, both pink and purple. To confirm that the presumed coliforms are not E. coli and to avoid any false positives due to oxidase-positive bacteria such as Aeromonas, the pink colonies are selected and the oxidase test is performed. A positive reaction would be the appearance of a dark color or almost immediately within 30 seconds. This reaction will not be observed in coliforms as they are oxidase negative. So we continue with the following indicator. ISO 7899 describes the method for the isolation of several species of intestinal enterococci, including those of greater relevance, such as Enterococcus faecium and Enterococcus fecalis. The basis is very similar to the previous one, which is the sample is filtered through a membrane filter, and then this filter is placed on a selected medium. In this occasion, the slanted spotly. This medium has sodium azide in its formulation, which helps to inhibit gram-negative bacteria, being, of course, Enterococcus gram-positive, and also TTC, which is a colorless compound, but due to the action of enterococci, they reduce it to formacin, which has a reddish color. Because of it, typical colonies of enterococci in slanted barley, as we see on this image on the left, have a red, brown, or could be even pink, depending on the species, color, either in the center or throughout the colony. Then these typical colonies are confirmed on bile esquilene acid agar that has been preheated at 44 degrees Celsius. Enterococci are capable of hydrolyzing esquilene at this temperature in a couple of hours, and they form a brown to maybe black compound that diffuses in the same medium, as we can see here on the plate on the right, and this is taken as a positive reaction, and they are listed as intestinal enterococci. Here in the lower image, we have a close-up of colonies of Enterococcus fecalis on what would be a bile esquilene acid agar plate. So moving on, the next microorganism is Pseudomonas aeruginosa. Again, the membrane filtration of the sample is the first step, and this membrane is placed on Pseudomonas CN agar, which is selective due to the addition of cetrimide in its formulation and the nalidixic acid that suppresses the contaminants such as Klebsiella or produce. The characteristic green-blue colonies are from the pyocyanin production and listed as confirmed Pseudomonas aeruginosa. On the other hand, the rest of the colonies are examined under UV light, and the colonies that give rise to fluorescence are counted as presumptive Pseudomonas and later confirmed on acetamide broth. For the ammonia production. We will review it. On the other hand, any reddish colonies without fluorescence are also listed as presumptive Pseudomonas and confirmed by oxidase tests, then again acetamide broth and King B medium fluorescence. So for the confirmation step, these presumptive colonies are subcultured onto a non-selective agar. Pseudomonas is oxidase positive, so a blue-violet color has to develop. Then on King B medium, the colonies are incubated and examined under UV light, and any fluorescence in the course of incubation is recorded as Pseudomonas aeruginosa. Here in this plate, we can see the growth of Pseudomonas aeruginosa on a King B medium plate. And for the acetamide broth, the Nessler reagent is added to observe a characteristic yellow color, which would translate as ammonia production and confirmed as Pseudomonas aeruginosa. To summarize, the total count would be all colonies with blue-green pigmentation, that is the pyocyanin production, that are oxidase positive, that present fluorescence under UV light, and that are ammonia producing. So you have in this table the steps and the confirmation steps summarized. So the next indicator that we will review today is Clostridium perfringens. This microorganism is very interesting since it exists in the intestine as vegetative cells and in spores that we all know are very resistant to certain conditions. So they can remain in water for a long time and also more than some of the enteric bacteria that we have reviewed. Therefore, its presence may indicate remote or intermittent fecal contamination. In addition, spores are not always inactivated by common disinfection processes, so they are a valuable indicator for evaluating water treatment. In this case, a thermal pretreatment of the sample must be carried out, heating it to 60 degrees Celsius in a water bath for 15 minutes. Here, it is very important to monitor the temperature, since the time necessary to reach it should not exceed this time. Then the sample is filtered through a filtration membrane to retain the spores. This membrane is incubated anaerobically on TSC agar, which is a selective and differential medium. Cycloserine inhibits the accompanying flora, especially Bacillus, and both ferric ammonium citrate and disodium disulfide are indicators of hydrogen sulfide. In addition, the incubation temperature increases the selectivity for Clostridium perfringens. After incubation, the suspected colonies are enumerated, since due to the reduction of sulfide to sulfide, it usually produces colonies of a black, gray, or even brown color. This black coloration of the colonies disappears quickly, so it is very important to count within the 30 minutes after the end of the anaerobic incubation. For confirmation, the ISO marks that all colonies formed should be confirmed, and if there are more than 10 colonies, at least 10 should be chosen at random. The medium for this could be blood agar that we see in this image on the right. However, you can also use some other medium rich in nutrients such as a TSA or a Columbia agar, and these must also be incubated anaerobically. The acid phosphatase test is performed on the colonies obtained from this subculture, and if a violet color develops in the first 2 minutes, as we can observe in this small image on the left, then it would be considered a positive reaction for the presence of acid phosphatase, and therefore considered as Clostridium perfringens. And finally, we move on to the last method, very simple one, ISO 6222, for the enumeration of culturable microorganisms. That is, all aerobic bacteria, molds, yeast that are capable of growing on a nutrient agar, that is the yeast extract agar. On this occasion, the membrane filtration method is not used, but the pour plate method, which was one of the other techniques that we saw earlier for quantitative counting in water analysis. So we put the sample in the Petri dish and then add molten medium, and we mix with a gentle rotation. Then the medium must be allowed to solidify, and then incubation at least 1 plate per incubation temperature. After this, the plates are examined and observed colonies are counted. So it is rather simple. In general, it is safe to say that ISO methods are simple. However, sometimes a couple of media are required, either the first for primary isolation, the second for subcultures. So let's see what quick solutions we can find available at Condelab. We know that traditional methods involve a lot of time, a lot of effort, and that is why the market demands techniques that meet this need of results in a short period of time, but using reliable methods, of course, with a high performance and also a high sensitivity and specificity to avoid any false positives. The techniques available are more or less established at the moment, range from molecular diagnosis, qPCR, which is fair to say the most used technique, to immunological tests, MALDI-TOF, or even NGS. These, of course, offer multiple advantages. However, the biggest disadvantage is the obligation of a highly equipped laboratory and also the specialization of the personnel to be able to carry out the tests. And of course, some equipment can be worth thousands of euros, so the initial investment is quite high. In this sense, our Condachrome line is an intermediate solution which provides all the technical advantages and can be easily adapted in any type of laboratory, even the same one in which the traditional methods are already being carried out. It does not require an investment in specific equipment, and the same personnel can use these media without any problem. The main advantage of Kondachrome is that anyone with previous experience can identify and see the results with the naked eye at the end of the incubation time. This is possible thanks to the Kondachrome substrates that give each microorganism an unique and distinguishable color, and in fact, in some media, different microorganisms can be observed on a single plate. This, of course, translates into time saving and also space saving. In addition, the know-how that have been given to us because of our 60+ years of experience provides our Kondachrome line to have an excellent performance and a very high sensitivity and specificity. So focusing only on water analysis, as we have seen, some chromogenetic media have been included in ISO methods such as the CCA, but for other indicators such as enterococci, we have two media available, Kondachrome MIE and Kondachrome MIE modified. With Kondachrome MIE in a single step, we will be able to identify enterococcus on our sample that will grow as a blue colony. If tetrasodium chloride is added, if you want much more specificity of the media, then the colonies will be red. And due to the selectivity of the medium, gram negatives will be inhibited, and so will all other gram positives that, of course, are not enterococcus. In the ISA method, two media are used. If we remember quickly, Slanets barley and then bile esculenacide agar. But using this method, simply the membrane is placed on the plate, incubated, and 24 hours later you have the results. On the other hand, modified Kondachrome MIE, which as its name indicates, parts from the previous media, this allows to differentiate between Enterococcus faecalis and Enterococcus faecium. The first will be greenish colonies, while the second will grow as deep blue colonies. Finally, we have Kondachrome pseudomonas. This medium is temporarily unavailable because it is currently under development for improvements, and some of our beverage company clients will internally validate the samples as soon as it's ready, because this genus of bacteria has a great impact not on the final product per se, but rather is an important indicator in the control of production water. In this case, as characteristic of Kondachrome line, the plates are easy to read. Pseudomonas will present magenta colonies, and since it is a medium with a great inhibition for the rest of the microorganisms, any accompanying flora growth, if it were to appear, it would be as colorless colonies. Again, in 24 hours, you will have your results. In all of these cases, enterococci or pseudomonas, the membrane filtration method is used. So the filter is the one that is simply placed on the Kondachrome plates, and shortly you will have your results. Finally, we will present very quickly the products that we have available. Our catalog, of course, includes all the media and diluents indicated in the ISO, and the Kondachrome line as well for a fast and reliable response. We have these available in a wide variety of formats, both in dehydrated and ready-to-use media, of course, in water plates. That is the interesting format. So with this slide, we're going to conclude the webinar, and we will very quickly move on to answering some of the questions that have been sent to us. So let's see. Yes, The webinar, as usual, is going to be the recording of the webinar is going to be sent tomorrow, together with a certificate of participation, of attendance. So you will have it available, don't worry. Then on to some technical questions. In the membrane filtration method, is it possible to do the filtration without vacuum? I think you're referring to just letting the sample pass with gravity, I think. So of course it is possible. However, I'm sure the bacteria count would be affected by the sedimentation time, or even solids remaining on the surface of the funnel because of the prolonged contact. So our recommendation, of course, is to follow the ISO method and use a vacuum. Okay. Is Condachrome media now, is Condachrome media ISO validated? Okay, so I think you mean that if the Condachrome products are accredited by an organization, in the sense, no, but they have been validated internally and also by clients that are reference in the beverage industry. They are manufacturers, multinationals, and they have validated the detection methods using the products of Condachrome line, because they really gain days compared to the ISO method. So they have found it much better method, if equally or even better the performance. So the internal validation would be the way to go if you want to use them. If now, for example, you have the ISO method implemented or any other alternative method, but you want the benefits of Condachrome. When growth appears all over a filter, it means that it is contaminated or just a colony that grew that way. I think you refer to growth around the membrane, in the border, right? So in this case, when you observe growth at the edge of the membrane, surely it is a problem again of the vacuum that has not been done well, and therefore the filtration is not homogeneous. This can be partly due to a poor closing of the funnel or a failure of the vacuum apparatus. So you have to investigate what caused it and correct it, because this is not right. To do an analysis, it cannot be done. Let's see. Do you have any media for coliphage detection? Yes, we have one of the ISO media. There are 3, we have one. However, because of this new directive that is going to be in place, we are, of course, thinking about developing the rest of the media. So if you are interested in coliphages and the method and the analysis, please send us an email so we can review the development of these media. We need to find out the interest that is out there, so we can do it well, earlier or later. So yeah. What alternative methods exist for water analysis? The most effective method, of course, is chromogenic media, which is our Condachrome line. That is being gradually introduced in ISOs. It is, in reality, it is very difficult for other methods to exist, because the disinfection of water, of course, can have an effect on the viability of the microorganisms present. So, for example, by using a molecular technique, you risk an overestimating the real concentration of any contaminants, because of these viable cells are the biggest obstacle to the implementation of any of these molecular techniques. So, of course, further development is required to standardize them and make them applicable in a variety of matrices. Of course, there are alternative methods on the market, it is true, but an internal validation must be performed. So, and what is more interesting in the water analysis are the analysis of indicators rather than pathogens, which we can see again in the somatic coliphages case in the new directive. Okay, people are asking about coliphages. We will send you the information. And okay, we have other questions, but I don't want to take further because I don't want to take more of your time. We will answer them by email, don't worry, during this week. So once again, we hope that this webinar has been interesting for you. Again, the recording and the certificate of participation will be sent tomorrow. We thank you again for joining us in our CondaLab talk, and you can write an email at micro@condalab.com. We have, of course, one more webinar planned for December. I don't want to give too much information, but it will be very interesting, and there might be something new from Conda. I don't want to say anything more. We will also be in Medica in November, so please, if you attend, don't forget to visit us at Hall 1, stand A45. So as well, all the information will be posted in our LinkedIn and also will be sent through our newsletter. So don't forget to follow us, to subscribe to the newsletter. And that's it. Without any further ado, let's see you in the next CondaLab talk.